All right, so now we're going to talk about peptides. Peptides are what happens when you link amino acids together. Remember that every protein in your body is made up of these 20 amino acids just linked together in different ways. So proteins are peptides. Truthfully, contextually, when they're tiny, we tend to refer to them as peptides, and when they're large, we tend to refer to them as proteins. But the real name for a peptide comes from the bond that's formed that links those two groups together. Remember, every amino acid has that amino group, which is actually an ammonium group, and carboxylic acid group, which is actually a carboxylate group. These can form a peptide bond with one another, right? So that's how amino acids are linked. The amine group and the of one forms with the carboxylic acid group of the neighbor. Nomenclature here. So when we have 2 amino acids, we refer to as a dipeptide. 3 amino acids is a tripeptide, 4 is a tetrapeptide, 5 is a pentapeptide, and anything longer we just call a polypeptide. Okay. And again, where does the name come from? That's the name of the bond that's formed. If we were in organic chemistry, we wouldn't call it a peptide bond. Well, our peptide bond, we would say, is made up of an amide group, right? Remember, even before we started talking about proteins and we learned that carboxylic acids will react with amines to form amides. And so that's what's happening here. You have the carboxylate group of one amino acid reacting with the ammonium of the other amino acid and forming a peptide bond. This doesn't just happen spontaneously. There's another protein in the cell whose job it is to catalyze that reaction, meaning there's a protein that grabs amino acids and sticks them together forcefully. Okay, the bond in the middle, the one between the nitrogen and the carbonyl carbon, is what's called the peptide bond. That whole new functional group that's formed in orange here, we know this from our study of organic chemistry. This is an amide group. And now in the context of peptides, this carbonyl carbon bound to the nitrogen, that is what's called a peptide bond. All right, so multiple amino acids are linked together through peptide bonds, hence the name peptides. All right, naming of peptides. We have this standard way that we write out peptides, and that is starting with the amino acid that has the free N-terminal group, and then ending with the amino acid that has the free C-terminal group. So what do I mean by that? Let's color code here. The N-terminal group would be the one that has the free ammonium group, not part of the peptide. That would be what's called the N-terminus. And then the last one in this that we refer to is the one that has the free C-terminal group. And by that, I mean the free carboxylate. Right, so this would be a linkage of 2 amino acids, glycine and alanine. We're going to list it as glycine alanine, not the other way around, because the glycine has the free amino group, the free N-terminus, and the alanine at the end has the free C-terminus. Okay, so there's this standardized way of listing the amino acids in a polypeptide. You go from N-terminus to C-terminus, never the other way around. All right, and if we wanted to name it, actually put a formal name to it, what you would end up doing is drop either the -ene or -ate ending of the amino acid and replace it with -yle. So for example, a tripeptide consisting of alanine, glycine, and serine, and actually let me put different colors here, alanine, glycine, and serine, where the alanine is on the N-terminal end, that becomes alanyl, glycyl, serine. So you don't drop the ending of the C-terminal amino acid, you just leave it as it is. Okay, but you list them from free N-terminus to the free C-terminus. All right, so this is an awesome problem here to practice with. We're going to identify the N-terminal amino acid, the C-terminal amino acid, and then name this tripeptide. And again, remember, you're not just expected to look at this and then go. You're allowed to use your constants page. So I would actually recommend everybody take a second and pause this video and see if you can't do this type of problem on your own before you watch me go through it. First, you got to identify where the linkage between the amino acids is. Like, where does one amino acid start and the other one end? Remember, that's at the peptide bond. And so these carbonyl carbon to nitrogen, these are our peptide bonds. So that means that this is my N-terminal amino acid. This is the middle amino acid. And this guy here is my C-terminal amino acid. Okay. What's the R functional group for my N-terminal amino acid? That's this thing here. So we go to our constants page, we're looking for that benzene ring. We got a few of them with benzene rings, but right, this one is actually a phenol group. This one's got this big heterocyclic nitrogen going on, and this is the one that's just the benzene ring. That's phenylalanine, or Phe for short. So our N-terminal amino acid is phenylalanine. I'm just going to abbreviate it. What's the next one in the line? It doesn't really ask us this, but let's just go through and do all of them. So this one we identified as Phe. The R group on this one is this here. So this is our thiol group. Go through our constants page. Let me get rid of these. There are a few that contain thiol groups. I missed the one. Methionine, it's not actually a thiol group, but it does have a sulfur in there. So which one are we looking at in our question? This is just one carbon attached to a thiol group. So it's not threonine, it's not methionine. So this would be cysteine, or CYS for short. Get rid of my markings again. So our second amino acid is cis. Oops. All right, and then lastly, what is our R group? This is just this baby guy here, the methyl group, and that one corresponds to alanine, right? So one of our non-polar groups, but the one that's just a methyl group is alanine, or ALA. So what's at the C-terminus? That would be alanine. All right. Actually, I'll phenylalanine and just alanine. Then this one was cysteine. I'm going to write those all out because I'm going to need it for when I write the full name of this tripeptide. Remember, we're going to list it N-terminus to C-terminus, and all we're going to do is replace this INE ending with YL. So this would be phenylalanine. No. Phenylalanine, not claiming that it's going to roll off your tongue, but that's how we would name it. Cysteine. Don't know how that happened. Cis, what the heck? Come on. Cystol. Did I spell cysteine right? Oh, I did. I don't know if the E's there. And then the last one would be alanine. Remember, you don't drop the ending of the alanine of the last one, your C-terminal one. So let's see. I guess they didn't put the E in there. That would not be a huge mistake. Remember, so cysteine, I told you to replace the INE. It did sound a little weird, which is why I left it off in the first place. But if you did cystal or cystill, that would count. Either one is correct on an exam. Okay, it does turn out that this one is correct. They usually don't include the vowel if it sounds weird. Not that it doesn't sound weird to begin with, but anyways, that's how we name our tripeptide. Truth be told, you only ever name really tiny peptides this way. If a peptide has like 20 amino acids, you're not, it's going to have its own little common name, not phenylalanine. Okay, and truthfully, more than that big, ugly, complicated name, what you would see is the abbreviations like this, right? So we could name this polypeptide as well, but if you were reading a scientific paper that would talk about this, you would more likely see it just as this right here with the 3-letter abbreviations, or actually what's even more common is like ALCM, a list of just the 1-letter abbreviations. Remember that each polypeptide has a 1-letter abbreviation associated with it as well. Leucine and methionine. So that's how I got these 1-letter abbreviations. So either of these would be like equally good representations of this particular polypeptide. We're going to talk about there's like different layers of structure to a protein. And so the simplest one is what's called the primary structure. And that is just a list from N-terminus to C-terminus of the amino acids in that protein. All right. These eventually fold in on themselves and they form different patterns. And that's the secondary and tertiary structure. But to a first approximation, we can say a lot about a protein just by listing the amino acids that make up that protein from N-terminus to C-terminus. And that's what we call a primary structure. All right. Yeah, so for example, the thyroid hormone releases this thyroxine, which is an amino acid sequence, glu-his-pro, also abbreviated EHP, because E is the 1-letter abbreviation for glutamic acid, H is the 1-letter abbreviation for histidine, and P is the 1-letter abbreviation for proline. If you wanted to draw it all out, we of course could, using all of those various structures. But yeah, this would be considered the primary sequence of this amino acid, just the list of the different the primary sequence of this polypeptide, just the list of all the different amino acids involved. Insulin is a really important signal protein, and this is actually pretty cool. Let's look here. So first of all, it's actually 2 polypeptide chains that are bound together by these. These are these disulfide bonds we talked about at the end of the last lecture. Remember, in the end of the last lecture, we talked about how they were bad when they're forming just randomly in your kidneys, but they are also positive, like constructive features in many proteins. Insulin would be one of them, where they're absolutely essential. It's the way in which these 2 polypeptide chains are kind of linked together is through these disulfide bonds. And we could characterize insulin by the just list of amino acids involved. So the primary sequence of this first chain, chain A, would just be the list of the polypeptides from N-terminus to C-terminus. Glycine, isoleucine, valine, glutamic acid, I mean, you don't have to know these 3-letter abbreviations, but anyway, this here just represents the primary sequence, the list of all the different amino acids from N-terminus to C-terminus. If you Googled insulin structure right now, you might see that actually this doesn't exist as a long chain. This thing will kind of fold back on itself and make a little bit more complicated of a structure. And we'll talk a little bit about those structural features in future lectures. We're taking it step by step, right? So again, the first thing that we can say about insulin is just listing those amino acids in order. Okay, so now it wants us to draw this polypeptide, this tripeptide, and give the name given the 3 amino acids that it contains. Now remember, the first one is going to be the N-terminal amino acid, meaning that's going to be the one with the free N-terminus or free ammonium group. So the first thing I'm going to do is start by drawing that N-terminus. That's connected to a carbon that's connected to the first R group. This one is for glycine or GLY. If I look on my constants page for glycine, I see that it's missing an R group, right? Here's my alpha carbon. Where's its R group? Well, just remember, this is just like organic chemistry. It has these 2 hydrogens bound. We just don't explicitly draw the hydrogens in there. Glycine is, again, that tiny amino acid, so I would just put hydrogens there. And then I have that carboxylic acid group or that carboxylate group. But now that I've linked amino acids together, I'm not going to draw, this would normally be an oxygen if I was just drawing glycine, but I'm not, I'm drawing a polypeptide. So the next thing that comes is that peptide bond or that amide group. So now I'm on to my next amino acid. So this guy here is glycine. I just want to, for the sake of it, color my alpha carbon, which is where the R group is coming off. So this is now going to be the R group for my next amino acid, serine. Going to my constants page, alphabetical order, I find serine. Serine has, here is your alpha carbon followed by 1 carbon bound to a hydroxyl group. All right, so my alpha carbon, then I have 1 carbon CH2 bound to a hydroxyl group. Remember, don't forget, the basics of organic chemistry that we learned. How do I know that there's 2 hydrogens there? Because carbon has to have 4 bonds. Likewise, my alpha carbon, all alpha carbons also have a hydrogen bound to them because carbon has to have 4 bonds, right? So keep in mind, we can't just forget everything from organic chemistry now that we're on to biochemistry. There's a reason why we learn organic chemistry first, so we can build. Now I got my, what was formerly a carboxylate group, but is now going to be this amide group for my polypeptide. So now this was my serine. And then I have my next alpha carbon. And again, color that just to emphasize these are the alpha carbons. That's what my R groups should be off of. And the last one was methionine or met. So I'm going to go here, find methionine, right? And so if you notice what I'm doing when I'm like copying these amino acids is I'm similar to before when we were talking about organic compounds, kind of just counting carbons to sort of make sure I got what's correct. Here's my alpha carbon, and then I got 1, 2, a sulfur, and then a third. So that's 4 atoms in a row where the third one is the sulfur. So 1, 2, have that sulfur, and then finally a methyl group on the end there. And of course that has to be a CH3 because carbon has to have 4 bonds. And then since this is the last amino acid in the chain here, this is the methionine, I'm going to have that free C-terminus or carboxylate group, okay? So just to emphasize here, The first atom in my list has a free N-Terminus. Everything in the middle has these polypeptide bonds. And then the last one has a free C. Whoops. C terminus. If I'm going to name my polypeptide, list your amino acids first. So glycine, it's probably a C. I guess I should double check. Yep. Glycine, serine, methionine. I'm going to drop the INE and replace it with an LY. So this will be glycyl methionine, right? For the last one, the C-terminal one, I just leave it as it is. Okay. Write the names and the three-letter, we're not going to write the names. We're going to just write the three-letter abbreviations for all tripeptides that can be formed between 2 glycines and 1 alanine. And actually what we're going to do is not even use the three-letter abbreviation. We're going to use the one-letter abbreviation because that's honestly what you will see 99% of the time if you go on to be a biochemist. So glycine, again, just to get in the habit of using our constants page, we're going to find glycine on our constants page. Whoops, got a lot of markings here. That one-letter abbreviation is just G. So this thing should have 2 G's. And then alanine has that abbreviation A. So one of them would be, let me actually clean this up because it's looking ugly. G, G, A. Remember, that's not the same thing as A, G, G. Those would be 2 different polypeptides. In the one, glycine is at the N-terminus and alanine is on the C-terminus. And on the other one, alanine is on the N-terminus and glycine is on the C-terminus, right? So those are not the same polypeptides. The last one would be GAG. That would be the third that you could make from those 3 amino acids. Okay. If we wanted to name them, just the naming kind of is a little bit silly, but nonetheless. So GGA would be glycyl alanine. AGG would be glycine. And then GAG would be glycine. Okay. You may have heard, there's like essential amino acids versus non-essential amino acids. This is something that you would have heard in the context of like dietary science. A non-essential amino acid is one that your body makes. So actually 11 of the 20 amino acids, your body can make all on its own. It can make it from carbohydrates and lipids and other sort of building blocks like that. The other 9, you actually need to have from your diet. Okay, so these would include, they tend to be the aromatic ones. So phenylalanine, tryptophan, I guess tyrosine isn't on here, and that's kind of interesting. Those can all be made. Yeah. I don't know that there's really any rhyme or reason to which ones can and cannot be made. I'm not going to really expect you to have memorized sort of the essential and non-essential amino acids, other than to say that 9 of them we can't make ourselves. Those would be essential amino acids. So essential, synonymous with humans cannot make. I wonder if we don't just, well, I feel like maybe phenylalanine gets converted into tyrosine. It's interesting that we would be able to make tyrosine, but not phenylalanine. But I trust the scientists that have established this list. Yeah, so anyways, that's why protein is such an essential part of just any diet. You really couldn't survive on just lipids and carbohydrates. If you were stranded on a desert island with nothing but sugar water, you really wouldn't be able to survive because your body would be able to make 11 of the amino acids from those carbohydrates, but not the other ones. So that's why protein is such an essential part of diets. There's also, if you're a vegetarian, you sort of have to be conscious about how much protein your body's intaking. And actually, even more than that, if you get back down to the nitty gritty, they have to be careful. I'm not a vegetarian, but I know plenty of them. They're careful about where their protein sources come from. So you have to have sort of a mixture of protein sources. You can't just have beans or grain as your protein source. You have to have a mix. And so actually, I believe that that covers the bases. If you have like a hearty grain diet and hearty beans in your diet, those cover all the bases, but you can't just choose one or the other because you won't get the full slate of essential amino acids.